Okay, so from beginning once again. So rheumatism is having two different aspects. One is an infective aspect, another is an autoimmune aspect. Okay, so both the things we should discuss. Also in practice, what you will see is these are handled differently. But since there are some relatedness, we must discuss it. One is called rheumatic fever, another is called rheumatic arthritis. Since the rheumatic terminology is there, we will discuss both. So the infective aspect is caused by a bacteria which is known as Streptococci, a specific class or specific species, that is Streptococci pyogenes. Now Streptococci pyogenes is a facultative anaerobe, and it is also a human pathogen. Usually different route it enters into the body. If we do a pure culture analysis by staining, we shall see that it is gram positive. We shall write it. It is gram positive. What do you call this? Gram positive. Kidhar gaya? Okay, gram positive. What do you call round shaped bacteria? Okay. Now they remains in chain, and therefore the name strepto. Strepto in Greek or Roman is known as garland or something. Jo aap putting in this thing for as a mala. So what exactly happens is, whenever it enters into the human body, it produces an exotoxin. Streptolysin O is the most common one, which is an oxygen labile protein. It's a glycoprotein basically. Carbohydrate residues like sialic acid and then N-acetyl glucosamine along with proteins are present. Okay. Now this antigen, whenever it enters into the human body, it produces an antibody. So any antigen produces an antibody. The antibody's name is given. The nomenclature of an antibody is from the origin of that antibody. So since streptolysin O is the antigen, and the antibody will be known as anti-streptolysin O antibody. Anti-streptolysin O antibody. Now what is the role of it? We can determine whether a patient is suffering from ASO infection, Streptococcus infection, if the ASO content or ASO titer is more than 200 Todd units. Okay, that also I should write. So normal serum value of ASO, normal serum concentration, shall we say, will be just 1 second, is less than equal to, okay, less than equal to 200 Todd units. You remember, Todd units, okay, not international unit, it will be Todd units. So what exactly happens is, if anybody's ASO content, ASO means what? It's a serological test. What we do is we take blood, then isolate the serum, and then do an agglutination assay. Now if in the agglutination assay the test is positive, we say that the person is ASO positive. Okay. Now what will happen? If the ASO positive is there, it will be at least within 200 Todd units. Now if it is more than 200 Todd units, it should be treated. The patient should be treated, usually by what? Penicillin injections. Okay. So beta lactamase. Remember, beta lactamase. Okay, beta lactam antibiotics. Mostly, by and large, beta lactam antibiotics regime. Okay. So what happens if a person is suffering from this particular bacteria? What will happen? He'll definitely have the ASO, what you call, what? Anti-streptolysin O antibody in the serum. Now if the serum is positive for ASO, it means that the patient is suffering from this condition. Now what is the outcome? If a patient is suffering, what is the outcome? Let us discuss this also. Then we will enter into this, the autoimmune aspect to it. This particular bacteria is beta hemolytic. Why this is beta hemolytic? Because if in a blood agar plate, now blood agar is what? It's an enrichment medium where nutrient agar is there, and along with that we use heparinized blood, not human blood. It is usually cattle blood. I do not know how you are having this particular information with you or not. Now what happens? See this. See, this thing happens. For example, you are having a particular what you call pure culture. Okay, let us draw it here. बार बार ना ये चेंज करना पड़ता है, बहुत ही दिक्कत है. Okay, so consider this. This is a test tube. Okay, I'll make it a test tube. Then this is a test tube and we are having a pure culture of Streptococci. Okay, consider that we are having a pure culture of Streptococci, something like it's a slant. Okay, it's a slant. Now this is called pure culture of Strepto. Okay, pure culture of S pyogenes. Okay. Okay, now what we'll do is we'll do a subculturing. Subculturing का मतलब क्या है? We'll put this into the blood agar. Okay, so how we'll put it? We'll put it like this, that it becomes a small colony. Okay, it becomes a small colony. Consider this. This is a small colony from a single bacteria. It is developing. Okay, small colony. Now there will be an interesting thing here. So remember, blood is there. Okay, now a very interesting thing will also happen. What happen is, okay, what will happen is, around these colonies there will be a white patch. Okay, now this is called zone of hydrolysis. So what is getting hydrolyzed? Who can tell me what is getting hydrolyzed? Blood is getting hydrolyzed. Okay, so we'll call it zone of hydrolysis. Okay, what we'll call it? We'll call it zone of hydrolysis. Zone of hydrolysis. Now the what we call is the radius of this hydrolysis is directly proportional to the amount of bacteria which is present in a single colony. Okay, leave it aside. Now for this particular reason we are calling this particular bacteria as what? Beta hemolytic streptococci. Now what is beta hemolysis? Beta hemolysis is a kind of hemolysis where a complete clear zone will be seen around the growing organism, because the growing organism has the capacity to destroy the RBCs and use the content for its growth. Understood? So as it increases in number, the zone of hydrolysis also increases. The radius of zone of hydrolysis is proportional to two things. One, how long you are incubating the organism in the incubator, or how much quantity was present in the starter culture. Okay, now the quantity of starter culture is calculated by a thing called CFU, colony forming unit. So we are not discussing that. So this is the case. Okay, so this is the case of this. Now how this is how it causes these problems. Okay, this is also very important. We will come to this particular point now. Okay, so what are the long-term complications? Long-term complications of ASO, as you all know, most of you. So in long term there are several complications of this ASO infection. Okay, first and foremost, take number is glomerulonephritis. Okay, then you can ask me, sir, why this glomerulonephritis takes place? So this is very interesting. We'll come to this point. Then rheumatic. Now this glomerulonephritis is also a rheumatic condition only. I'll come to that point. Rheumatic heart disease. Okay, these are the most common thing that is seen in India. Okay, the long-term complications. Now why this thing happens? Okay, so in this case we will draw this. Just one second. Okay, so what exactly happens is, so this is the whole thing as we have discussed till now. We'll come to the autoimmune condition later on, a bit later.